So maybe I will start with just a little introduction to the CFD modeling. So can you go to this next slide? So computational fluid dynamics is actually, what does it do is actually modeling a technique which is based on the real physics. The equations which are used are the real physics behind the flow. And there are normally no trial and error methods. So these are like the true equations based on which we do the simulations. So you always start with either an existing or even a non-existing system, even a hypothetical system, and you transform that into a digital or a computer-aided design of your system, of your bioreactor, for example. We then transform this into very small volumes, which we call a mesh, where we solve all the equations. And based on that, we solve it, we model it, and you get a nice output, which is normally in 3D, which means that you could see it along the width, along the depth, and in any dimension, and which is a function of your variables. But the real power of such a tool comes when you actually run different scenarios, what-if scenarios, which is actually very important given we want to figure out what is the best mitigation strategy for N2O. And you can run multiple scenarios. For example, you can change your aerator configuration and see what happens. So you get a new input, and you can compare it, and you can make an assessment which is the better design amongst all the what-if scenarios. So this is like a basic understanding of how CFD works. But what we do actually now is that to really get to the depths of the problem, which is the N2O emissions, we add kinetics into it. So kinetics is actually coming from these IWA scientific reports, which are very famous or very much used in the industry. Activated sludge models, ESM1, and etc. We combine them with the CFD, and then we try to predict the concentrations. And in the figure you see actually is a treatment facility where there were very high nitrates coming into the system, and there was actually a small mixer at the bottom, which was completely short-circuiting the whole nitrate stream towards the outlet. You would not be able to see that if you would only purely do the CFD modeling. You would see, okay, there are streams coming, but how much is it? How bad is it? You want to know that. By using CFD and kinetics together, you can understand the impact of your design on the performance of your biological system, and then you can run scenarios by moving your impeller or your mixer somewhere else and see if it actually improves your system or not. Okay, so that was me. I think now it goes to Ingmar.
Yeah, thank you, Usman. So what else can we then learn from these CFD models coupled with kinetics? So we have already seen that you can basically estimate local concentrations of all the state variables that are in the ASM models. And that allows you to evaluate your effluent quality, but also to evaluate energy optimization. For example, if you play with the airflow rate, you can run different scenarios. But as Wim already pointed out, there's a lot of criteria. It's really a multi-criteria problem. Which means that now when we add N2O and the mechanisms of N2O on top of this, if that is the primary objective, basically you will also get an insight in the other, both in the energy optimization and the effluent quality, and also be sure that this is maintained. So it's like killing 3 birds with 1 stone. And moreover, it also is kind of an X-ray of your 3-day X-ray of your system, basically turning it from a black box into a more gray box model, and seeing really what is happening under the surface. Now, a little bit on N2O kinetics, not the idea to go into too much detail. Obviously, they are not present in the traditional ASM models that have been published earlier in the STR that was mentioned. But basically, model extensions have been developed over the last decades. And have been putting in pathways and have been calibrated typically at lab scale. And so what we did is we actually took the most important pathways of these model extensions and actually added it to the biokinetic model, so both in terms of stoichiometry and process rates. And that basically allows you then now to make local predictions of all the relevant species that have an impact on the generation of N2O and also of the N2O concentration itself. And yeah, if new kinetics would be further developed, they can easily be added, just like adding more mechanisms and species in a Guyer matrix, as we are typically familiar with. One thing that I would like to stress is that you should also mind not over-parameterizing your model. So you can add a lot of mechanisms, but at some point, yeah, if you have limited data available, it's very difficult to calibrate those models. Okay, let's go to another poll here. I will quickly introduce this system. So this is actually a reactor where the inlet is on the bottom left and the outlet is at the top right. And first you have an anoxic zone with baffles, and the arrows indicate how the liquid is moving. And then the final part of the reactor is an aerobic zone. Okay, and so the poll question is, where would you actually start measuring? So would you measure at the start of the anoxic zone, at the end of the anoxic zone, or in the aerobic zone? Would you have a preference where you would on how you would design your measurement campaign? Yeah, so you also have the option all of these locations, or I have no idea. That's also a fair answer, of course. Okay, let's have a look at the results. Okay, so yeah, okay, so the winner is all of these locations. That's obviously always, let's say, conservative or a safe option, because then you will get to know everything. But then, yeah, interesting. The others, like the aerobic zone, is also quite popular, and then at the end of the denied zone. Okay, interesting. So let's then continue and actually also give a little bit an insight in what actually comes out of this model. So here you can see the N2O production rate in the different zones. So we are now looking at the top view of the reactor. So it's like a horizontal slice. And left is the anoxic zone and right is the aerobic zone. The base case is a base case with a certain airflow rate. And here what we can see is that actually in the anoxic zone there's not too much going on with respect to N2O production. In the aerobic zone, it's also fairly limited, and it's actually at the end of the anoxic zone where we observe quite a bit of N2O production. And the reason actually for that, you cannot see it from here, is because actually there's a backflow of oxygen from the aeration zone into the anoxic zone. Okay, so clearly a design issue that is causing this. In the middle row, middle figure, you can actually see that now it's a different scenario where actually the airflow rate has been increased in the aerobic zone. So you can see that actually this further emphasizes or increases the N2O production in this anoxic zone. And the lower figure is again at the same airflow rate, but now at a different load, also illustrating that the load has also an impact on the N2O production. Okay, and then from these simulations, you can have the observations of not only the N2O, which I discussed now, but also you can see when you, for example, reduce the aeration rate, what the impact is on, for example, the effluent ammonia, to keep an eye on that, and for example, also on the nitrate concentration. Okay, and you can see that, that they can be very different in terms of N2O production, even if your effluent is not really affected by the changes that you have made. Okay, and this is another example, again, from the Eindhoven case. So here you see 3 measurement points, so 1, 2, and 3 indicated on both figures. And the difference between the top and the bottom figure is, again, the same simulation, but with a different influent load. So meaning that if you look at the figure on the right, we are just looking at two different instances of the same day. So it's actually a loading cycle that comes in. And then from the measurements that you see there, you see a certain sequence with respect to the amount of N2O that is produced at different locations. And you also see that that differs throughout the day. And actually, if you look at the colors in the figures, you can also actually tell that the predictions that are being made are qualitatively in conjunction with what we have observed, including also the fact that the red zone, where you have the largest production, actually moves inside the tank, this volume that is indicated there, and that basically it becomes quite cumbersome to keep an eye on that. So the next slide, I think, brings us to the third key point. So the CFD with the integrated kinetic model, including the N2O kinetics, is a powerful assessment tool. So you can actually observe or evaluate where in your reactor and to what extent certain N2O is being, concentration is being produced. And it could also help you or point in the direction where you have to look for mitigation strategies. Okay, so the CFD-Biokinetics visualizes actually the concentrations in 3D as a function of the plant design and the operational conditions. It's, as I said, kind of an X-ray from your system.